So on the IB exam, you might be expected to look at micrograph images like this one and then be able to say what that is. So we got to go through these and practice them, and we'll do some of these in class to, during warm-ups to flash up these images and see what you think. So here, free ribosomes, all these little small spheres, they're very tiny, even electron micrograph, they're really tough to see. So that's a tip-off you're looking at. Just ribosomes. So ribosomes, you don't see any membranes associated with them. And remember, ribosomes are always making protein. They can either be on the ER or free. So these ones look like they're free. And right here, lysosome. You see, it's just a dark. They appear dark. You don't see much inside, and they're surrounded by a membrane. They pop out of the Golgi. And they have digestive enzymes help to break down waste and any parts the cell doesn't need anymore, like a garbage system. Break it down, reuse the parts. Vacuoles vesicles, we can see this is a plant cell. Usually they take up kind of the middle, and usually they hold water, waste products, extra food, things the cell might be storing. So like a storage structure for a plant cell. And only a large central vacuole, just the plant cells have. So maybe make a note of that. Large central vacuole, plants only. And here we can see the nucleus. Again, the membrane surrounding it. This dark stuff here would be chromatin. That's the word for DNA plus the protein that's associated with it in eukaryotes. And here, actually I should probably chromatin here. This dark structure here is nucleolus. That's one way you can tell it's a nucleus, not something else like a mitochondria. You see that dark circular structure, and this is where ribosomes are made. Ribosome, kind of hard to see. And here, cilia and flagella, very cool structure. If you count these, you'll see 9, and then there's 2 in the middle. So it's called this 9 plus 2 arrangement of these little microtubule bundles. So pretty cool. Anyway, cilia are smaller. Ooh, not flagella. But either way, they keep movement. Cilia inside your throat right here, or inside your lungs here, the trachea. You have cilia that's pushing all the mucus back up. Mucus that got stuck with all the dirt and everything else that might be in the air that you breathe. So that gets kicked back out. So you always got fresh mucus, purifying, filtering the air you breathe. Mitochondria, telltale, they look like a jelly bean, but not necessarily B-A-N. They could look circular. That could happen too. If you cut it this way and look on this end, it'll look circular. But if you look, this is really cool. I really dig this electron micrograph because you really see all the folding of the cristae. There, it's spelled out a little better. Yeah, all you see these folds right here, all the membrane. That's cristae. And Golgi, I like looking for Golgi. You don't always find them. When we do, I think they look like a stack of pancakes. They're kind of there, and they help to process protein, package for export. Chloroplast, this is like a freeze etch technique. It's almost like a 3D image, but you see there's the outer membrane, and then you have the thylakoids, little stacks. It's kind of hard to see in this one. And here are some microtubules. You'll see here, and they help to move things along, move the chromosomes in mitosis and meiosis. And then the ER, and this right here looks like it's rough. So rough means it has ribosomes sticking to it. And ADS for eukaryotes. Maybe make a note of that. And if you remember prokaryotes, you remember what they have? They have 70S, slightly smaller. S is silvert, I think I remember that right. Anyway, it's just a unit. You don't need to know anything about it. So we can label our prokaryote here. These are pili. Okay, these little fingers coming out. The nucleoid, or just the DNA. You know, that's long circular. It's just one big circular piece of DNA. Boom. It's like circular, but then it's twisted up. And these little dots, those are ribosomes. So they share that with our, but remember they're 70S. And the very inside is the plasma membrane. And right outside is the cell wall. And the very outside is the capsule. And you can see these long things here some bacteria have for movement, flagella. Okay. And you can practice measuring this and then dividing to get the magnification. Why don't you do that just to test yourself? Okay, I'll leave that to you. And this is pretty cool. This is pretty extensively labeled. Picture diagram, you know, the cell. Remember, everything is kind of idealized here. And some of these parts you don't really need to know about, but you know, starting in the middle, you know, the nucleolus right here, the nucleus, just labeling it chromatin as that dark stuff. 4 is kind of hard to see, but it's right here. It's a nuclear membrane, maybe a pore. I'm not quite sure what it's pointing to. 5, it's also fun to try and find. Oh yeah, 5 right here. So you see, look like a jelly bean, and then the cristae. I'll just always remember that. 6, oh yeah, here we go. All the membranes with the ribosomes, you know, the ADS ribosomes surrounding it. And that's what 7 is. Where's 7? Where are you at, 7? Where? Oh, oh my God, 7 right here. Yeah, ribosomes. Golgi, my beautiful Golgi, my pancakes, love pancakes, smooth ER. 9, that's another toughie right there. You just like the ER, but without the ribosomes. So you're not making proteins, you're modifying proteins, processing toxins, making lipids. You get the picture. Peroxisomes, they got peroxide H2O2 in them, which some seniors now are doing their IAs on hydrogen peroxide. Lysosomes, 11, right here, and that's more likely they'll ask you about, you know, be able to, you know, identify. 12, this would be a bile capillary. This is technically a liver cell. So bile that's produced by this ER can be exported. 13 are desmosomes. They help to attach cells together, help communication between the cells. And then you got some microvilli for 14.